Okay, guys, I want to start by taking a quick poll. I was just having an interesting conversation with another professor who was asking me if I thought recitations were valuable and if we should do away with recitation. And I said, well, I can guarantee half of my B students would fail without recitation. How do you guys feel? So who thinks recitation is valuable and that you would probably do worse without it? Right, you have to commit. Okay, 23. That's probably about half the people here. So, who thinks? So what someone has proposed is, rather than completely doing away with recitation, making it available only to a fraction of the class. So those who want it can register for it. And I thought that just wasn't fair because no one wants to be in class extra time. But to know that some of your fellow students have an advantage just seems unfair in a way. Who sees recitation as an advantage? Who feels that it would be an advantage if let's say only 50% of the class could take recitation? And they would feel like it wasn't fair? Huh? Okay, who feels that they sit in recitation, it's a waste of their time, and wish it was gone? I heard someone say yes. I'm not going to judge you. I'm going to judge you. Okay. So you guys are, and those who are raising their hands are getting A's anyways? I'm just kidding. Okay. Okay. Huh? I'm not talking, I'm against making it optional, but it has been something that's been talked about. So we were talking about what would make the most sense. Yeah.
You know, you said like it wouldn't be fair for some of the students. Like, couldn't they do like you sign up and then if you want to do the recitation, you can be in the recitation. Otherwise, you just don't have to do the recitation.
Well, that's what it was going to be. It was going to be like 50% of the 50% is of the class enrollment would be able to get into recitation.
Oh, I don't agree with that. I think it should be everybody's allowed to, but you don't have
Because
to.
then, what if you can't get into like you're
Yeah,
like,
I know.
I got in, but I didn't get into recitation.
It's hard. These are the debates that we have amongst the
They make
faculty.
it optional, like where you could sign up, the whole class could sign up if they wanted to, and then if you didn't want to, you wouldn't be penalized for it. That's fine, but you're gonna miss out on it. So, oh well.
Right.
But it's your choice.
It probably would be less. It would probably be 3 credits if you didn't have recitation.
If I had an option to sign up for it, I wouldn't sign up.
Oh, really?
Yeah.
But you feel it's helpful.
Yeah. I think especially stuff like we did last week or this week.
Yeah. Interesting. Okay. Okay, thank you guys for your thoughts. I want to bring them back to the faculty. Okay, does anyone else have any thoughts to share about recitations? No. Yes.
Why don't we do like actual biology labs?
Because we don't have the facility.
Alright.
Yes, there's actually a cell biology lab that's designed and waiting until there's a space and resources. Yeah, that would be the best. Yeah. Okay, actually, I took a cell biology lab with my cell biology in undergrad, and it was a canned lab, and we did the most horrible. It was like 201 all over again. So depending on how it's done, I mean, you guys may have found 201 to be useful, but usually when you're done with it, you want to move on and do bigger and better, not just like the same kind of like. Okay. So this is our last lecture with the cytoskeleton. Who's really sad to see the cytoskeleton leaving? It's not really leaving. As you've learned, none of it is ever done when it's done, right? So we're going to finish our conversation about. Okay, guys, hush, hush. We're gonna finish our conversation on how regulation of the cytoskeleton occurs in response to outside signals, how it affects cell behavior, right? Okay, so just a summary. These are the topics you better know, right, for your exam. Components of the cytoskeleton, proteins that regulate, proteins that organize the cytoskeleton, motor proteins, all those we did go over in recitation this week. So that's a good half of it. Don't think that's your study guide. Someone said, "Oh, it's great. I got a study guide." I'm like, there's still the other half of the class. And that's, how are motor proteins regulated, which you can easily stick into that picture that you drew, or chart. Actually, if someone just showed me a chart, some people don't do well with pictures, and they do better with words, right? Had a number of people say that to me, and so someone converted all the information into a chart. So that's another way to do it. So you can easily put in regulation by phosphorylation and how that all works. And then we started to talk about the cytoskeleton and cell behavior. So we talked about muscle contraction, and we've been talking about cell movement, and we introduced the Rho family of GTPases, and that's where I'm going to pick up. Right? These are the molecular switches that control actin polymerization in response to a signal. Okay, so I'm going to repeat the first few slides from last time just to kind of orient you to where we are. Right, so this family of proteins is involved in actin organization to generate cell polarity, so a directionality, in response to extracellular signals. So the best example is in cell migration. Right, the front and the back of a migrating cell have different structures. We talked about how the actin in the front is going to be what kind of actin form? Web-like. Web-like, involved with protrusions and moving forward, creating a leading edge, right? And and what's going to be in the back? Contractile actin, right? And so different motor proteins will be associated, right? So in the green, in the leading edge, you're seeing myosin 1. And then in the rear, which is contracting and scooching forward, is myosin 2. These Rho proteins are part of the RAS superfamily. So we talked about what RAS was and how there's a whole bunch of G proteins or GTPases that we've learned about in this class, and they're all similar in what? How are they all similar? Yes? They're molecular switches. What does that mean? Right, yeah, so you're not going to make me turn on and off the light again. Thank you. Right, so they turn things on and off, and when things are on, they're bound to what? Right, so when they're GTP, they're activating things. When they're GDP bound, they're no longer activating things. They're off. Okay. Okay, so remember this. GTP binding proteins, right? So when you have GTP bound, they're active. You get GTP hydrolysis. They're inactive, and then that goes away. And then a new GTP comes in, and they're active again. And that's, remember, the gaps and the gaps and all that good stuff. And the difference, so there are many many members of the Rho family, RAC, Rho, and CDC42, and they all affect actin in different ways. Rho activates stress fiber formation. RAC activates lamellipodia formation, so involved with creation of formation of a leading edge and movement. And CDC42 activates Philippodia formation. So what does that mean? So how are these Rho proteins affecting actin organization? So I'm saying they activate things. Well, what does that mean? What are they doing? Like, what are they activating? Yes?
Maybe ARP.
Maybe ARP, right? Maybe formins, right? So we learned about all these accessory proteins, and so now you need to bring this all together, right? So when you're activating something, that means you're inducing growth of actin in a certain conformation to do a certain thing. So you need to think about, well, all these proteins we've been talking about, which ones activate growth, which ones stabilize, which ones break things down, right? All that information, you need to integrate that into this thinking. So if you're activating formation of an actin web, you're going to activate what protein? ARP, what might you inhibit? Formins, right? What else might you inhibit? What else could be inhibitory? So often you'll activate the things you need, but you might turn off the things you don't need, right? So we're going to talk about this in more detail. Okay, so this we showed you the other day. WASP proteins are frequently activated by CDC42 and RAC. Activated WASP binds to ARP and enhances ARP nucleating activity, right? So it's going to activate something that's going to bind to ARP and enhance nucleating activity. I asked you if this was familiar, and everyone looked at me like I had 3 heads. And I got angry and ended class promptly. Well, and it was time to end class. But now you're prepared when I say, does this sound familiar? Okay, good. Who's like, what is she talking about? Does anyone have no idea what I'm talking about? Okay, good, because I beat you over the head the other day. Right, so this is what we did in recitation last week with E. coli and TIR. Green is E. coli. Red are the different proteins that are involved. Remember TIR, what was TIR? A receptor, right? That's the one that the E. coli injects into the host cell. And the whole point is to form this actin pedestal. Is it coming back? Coming back? I see people nodding. Good. Right? So then you get... and then it was all dependent on this NEC protein, right? So when you add an actin, when you stain for actin, you get staining. But in the absence of NEC, you have it, but not as well. With WASP, right? You have it in the presence of NEC. In the absence of NEC, you don't have it. And ARP3, you don't have in the absence of NEC. And so NEC was this protein that was required for recruitment of WASP and ARP and formation of the pedestal. Right? Integrated all into what we were just talking about. Everyone feel good? And so you could see that it's all dependent on NEC, and that is what causes actin pedestal formation. Okay, so RAC activates ARP and other accessory proteins that favor formation of actin webs. We just talked about that. We saw that in the E. coli paper, right? So this is how it works, right? So you'll have PI3 kinase, remember that's involved in signaling. So you'll have some kind of signal come in which will activate PI3 kinase, which will activate REC, or will also, and then there'll be this feedback, but forget about that. Basically, REC is activated, and then you have activation of WASP, ARP, and then you get that branched actin web formation. It will also activate some other kinase, which will activate filamin. Remember, that was that cross-linking protein, right? So these are 2 things that are very important for creating this web-like structure. The other things that it does is it activates some kinases that inhibit capping proteins, right? Because we want web growth, right? And then it also will inhibit MLCK. Do we remember MLCK? We saw it before. We've been studying really hard, right? This kinase that activates myosin, right? So it turns that off. Why? What is myosin involved in? Is it involved in the web formation? No. What was it? Myosin 5? I forget which myosin it was. Is involved in the contractile actin. So it's turning that off. We don't need it. Keep it off. Right? It also inhibits MHC, which I don't remember what that is, but that's all involved in decreasing myosin activity and forming less stress fibers, because you're not trying to do that. Anyone have any questions? Yeah. Okay, good. So then there's Rho, and Rho, this is a similar kind of network, right? So you got Rho, and Rho is going to turn on formins, and then you get the actin bundle growth, more stress fibers, right? And then it also turns on this Rho kinase, which will inhibit the MLC phosphatase. Remember, phosphatase takes off. Yep.
Is there any inhibition or activation of cross-linking proteins? Cross-linking? Would it do any inhibition on those?
Row. That's a very logical thought. So the question was, will it inhibit those cross-linking proteins? You mean the one like filamin, right? Right. So my guess is it might, because it's not needed. It depends on how it's regulated. If it might be something, it's very logical that it would turn it off. That's a very logical thought. And if you were a researcher and you were studying activity of Rho, you probably would take a look at that, right? It turns off this MLC phosphatase, right, because that removes phosphate groups. Remember, MLCK is the kinase that activates myosin involved in stress fibers. So you need it to be phosphorylated, right? If it's not phosphorylated, it won't be active. So it's logical that you would want to inhibit the phosphatase so that the phosphate groups aren't removed. It's like forward then backwards thinking, but you just got to follow it a little. And then it inhibits the limb kinase that regulates cofilin. Bless you. I mean, it activates the limb kinase that will then turn off cofilin, right? Because do you want cofilin to be active if you're making stress fibers? What does cofilin do? Someone raise your hand. I hear writing answers. Just do it. Put yourself out there. Take the risk. It destabilizes actin. So if you're making stress fibers, do you want cofilin to be active? No. Right? Cofilin's really big on actin web formation, where you're disassembling in the back and you're reassembling in the front. Remember that picture with the arrow and all the monomers follow the arrow? A nice conformation. Right? So it inhibits cofilin. So this is just how I want you to start thinking. So not only do you need to understand what all these proteins we learned about do, integrate it into function, okay? What is the cell doing? What kind of proteins are logical to be needed? And no matter what you said, like what you said before, it's logical. It may be right, may be wrong. I don't know if the research has ever been done, but if you wrote that on an exam with an explanation, it would be right because it's logical. But if you told me cofilin needs to be activated for stress fiber formation, I'd be like, where's the logic there? It's nowhere, right? Okay, any questions about this? Right, so this is signaling. Are we done with signaling? Did you think we were done with signaling? Are we ever done with anything? No, never. Okay, so polarized cell growth requires actin rearrangement. And that's activated by CDC42, another Rho GTPase. This is an example in budding yeast. So polarized cell growth, this means formation of an end, like a specific end of a cell that's going to do something. It's polarized. And here the example is yeast and how they respond to starvation conditions, in a way. When you have starvation, the yeast will secrete mating factors, which will then bind to a receptor, and you'll have signaling, which will result in actin rearrangement to form a bud. And that would be your polarized cell growth, right? So it forms a bud, and that's going to form a spore. Remember when did anyone ever learn about like yeast or bacteria and how when times get tough and there's no more food, you need to form spores because spores are very stable, right? Is that right? No, I'm thinking about something else. Sorry, totally thinking about the wrong thing. Sporulating in yeast is about mating. Anyways, the point is, I was like, wait a minute, that's totally wrong. You guys get to relive this when you listen to this again. Yeah. So when you need to mate, they need to make spores. And then you get activation of CDC42 in response to a mating factor, which then results in this budding, which is going to turn into a spore, and then you'll have mating. The point is, the shmoo. It's just called shmooing. That's called shmooing. There you go. A little fun for the day. So this is what happens. That's what the East people call it. It's in the textbook and everything. Who saw it in the textbook? Who saw it? There's an actual picture of the shmoo in the textbook. See, Zari saw it. Okay. Mating factor binds to, what kind of receptor is that? Yay, GPCR. Okay. Binds to GPCR, signal, activation of Rho. Okay. And then you're going to have activation of Rho, formins, and you'll get actin cable formation, which is actually just a kind of contractile actin, which is going to be involved in, I guess, pinching off. And then you'll also get WASP and ARP, and then actin polymerization because you're moving forward. So it's kind of a combination of the two happening. And then you'll also get, and then there will be a feedback. So that's one example of the effect on cell shape and movement. Next example is, I keep going back to white blood cells and how they like to chase bacteria. So this is the chemotactic movement of a neutrophil. We saw that on the first day of this section, where you watch the neutrophil chase the bacteria. So here, this is a pipette. That's a pipette kind of leaking some compound that is only found in bacteria. And that's the chemoattractant. And the neutrophil wants to move towards it. And you see, this is different times. This is time 0. And over time, the bacterial will move towards the chemoattractant. And this is dependent upon RAC-induced activation of ARP. But it's more complicated than that, as always. So RAC activation, let's orient ourselves. Here's your bacteria, here's your chemoattractant, here's your receptor, right? It's going to activate different G proteins, which is then going to activate RAC, which is going to result in actin polymerization in the front. This is all done through G proteins. Remember, these use a lot of second messengers like calcium and cyclic AMP and stuff like that are short-lived and localized. So that's how only the front ends up forming the leading edge. But not only that, but it also then activates Rho in the rear. And then you get the activation of formins and contractile actin and myosin activation. And so that's what results in leading edge in the front and the contractile actin myosin in the rear. And they actually both inhibit each other, which is how you end up with the separation on either side of the cell. Are we good? Okay, we are almost actually, done.
Is RAC a part of the Rho family?
Yep, RAC, Rho, CDC42, all part of the Rho family.
This looks like a separate.
So there's Rho and there's RAC. They're different proteins. Yeah. So when I mean family, thank you. When I mean family, I mean a bunch of proteins that have similar domains. So they all have GTP binding domains. They all have, they're all part of, actually, so there's the RAS family, and underneath the RAS family, you've got the Rho family. And the Rho family all are involved with actin structure assembly regulation. Yes?
Do you want us to know what RACRO and CDC42 actually do, or just know that there's...
I think as long as you understand, one is involved with actin web formation, and that's involved with leading edge movement, and one, So I think know that very basic part, but don't memorize these whole pathways. But you might want to understand them so that you can use the information if I give it to you. Yes, so it's worth knowing what RAC, Rho, and CDC42 do. Yep. Not too hard, just 3 things, right? Okay. Good questions. Okay, yes, it gets even more complicated. So communication between actin and microtubules. Oh dear. I've put those in separate boxes. You mean they work together? Oh. It results in whole cell polarization and locomotion. So this is just an example of the immunological synapse. So you've got your T cell. That's the one of your immune cells that likes to attack virus-infected cells. So we'll call this one, just for kicks, we'll call it this one the T cell and this one the infected cell. And here's your nucleus. Here's your, what's that? Centrosome, your microtubules, actin. And then what ends up happening is you get a signal. So there's some kind of signaling that results in actin polymerization that happens right over here. And that causes complete reorientation of your centrosome and microtubules such that it can actually position your Golgi closer to the other cell. Why would you want your Golgi to be closer to the target cell? So I guess to understand this, you need to understand that cytotoxic T cells, at least, deliver these hits of these secretory molecules that are toxic and kill the other cell, because you want to kill your virus-infected cell, right? So the rest of you can survive, the organism survives. So why do you want your Golgi to be close to the side of your cell that's interacting with the infected cell? Yes. You're always like, fine. I'll give you an answer. Yes?
Well, I mean, you're aiming. The Golgi is where everything gets ER, things get made, it gets modified in the Golgi and then secreted out of the cell, and so you just want to aim the gun, essentially.
You're aiming your gun, right? Remember, things that are secreted go ER, Golgi, secretory, vesicle, plasma membrane, out of cell, right? So if you orient everything so it's right up against the cell that you want to kill, it's much more efficient. Like, it would be really dumb to secrete it out this way, right? Would that be dumb? Would it ever make its way over here? No. No. And so this is all regulated in ways by, or not regulated, but RAC and Rho are all involved in the communication between actin and microtubules, and it's good stuff. Right, so RAC and Rho will also regulate different proteins involved in microtubule dynamics, such as Stathmin, right? What does Stathmin do? Stathmin destabilizes microtubules by doing what? So that's an indirect effect. By what? It inhibits them, but how? Yeah?
Doesn't it bind to subunits?
Right, so it binds microtubule subunits. So it basically lowers the critical concentration. So that does result in destabilizing microtubules, but that's like a little bit down the road, right? Because it's all regulated by concentration. So if your concentration is very low, the microtubules start to depolymerize. So if you inhibit Stathmin, then it will encourage stability of microtubules. And microtubules then also modify actin dynamics. So I'm not going to go into gory detail with this. You're welcome. But I do want you to appreciate the fact that it's all connected, right? So it's not like either actin or microtubules. It all works together to get the job done. And it's back to the big picture, all about the cell responding to the outside environment. Okay? Okay, I think this might be the last, this is the last slide. Oh, why do I always do it that way? Okay, so neurons, we're going to give a moment to neurons because we always ignore them. They're specialized cells that receive signals through dendrites and transmit signals through their axons to their target cells, right? So here's your cell body, your dendrite, your axon, that's a lot longer than that, and then your synapse. And so don't forget, you need things to move down the axon to get to where they need to go, right? So you have vesicles that walk along microtubules, and they bring things back and forth. And just like it's similar to how things move in non-neuronal cells, they're using microtubules and motor proteins to transport proteins, vesicles, organelles, mRNA, extracellular signals. All of this is happening on these microtubule tracks. And it's just important to understand that the axon's very long, and remember the microtubules have this long persistent length. And I think that's it. Okay, so we've got like 15 minutes for review. And I'm not saying that I'm doing any review, but if you have questions, now's the time. Because I know some people have told me that they might have questions. Otherwise, you can leave, but that means you're giving up 15 minutes with me.